Hello everyone, myself Dr. Parth Goswami, and today I am going to teach you a very important topic from neoplasia, that is a growth promoting metabolic alteration. So in the cancer, basically there are three forms of metabolic alteration. One is Warburg effect, second one is autophagy, and third one is oncometabolism. So all these three comes under the metabolic alteration in the cancer cell. Now let's see the learning objective for the today's lecture. So friends, this whole topic comes under the competency number 7.2 as per the Indian curriculum. And the competency of number 7.2 consists of molecular bases of cancer. So basically there are 8 fundamental molecular bases of cancer, one of which is growth promoting metabolic alteration that we are going to discuss in our today's lecture. So it could be the metabolic alteration is of 3 types as we have discussed: Warburg effect, autophagy, and oncometabolism. So let's see the growth promoting metabolic alteration in the detail. Now, before that, I want to quickly revise the 8 fundamental changes in the cell physiology, that is the molecular basis of cancer. The 8 fundamental changes occurs in the cancer, which is known by the name hallmark of the cancer. So there are 8 molecular bases of cancer. The first one is self-sufficiency in the growth signal. It means, there will be activation of the oncogenes and the oncogene protein products, will lead to cancer formation. The second important change is that resistant to the tumor suppressor gene in the cancer. The tumor suppressor genes are not functioning well. The particular example is p53 and the retinoblastoma gene, right? They will not work properly, and so that cancer is not prevented, and so the cancer will occur. The third important molecular basis is evasion of apoptosis. We know very well that the apoptosis will remove any abnormal cell if present in the body, including cancer cell as well. But in the cancer, in the fully developed cancer, they find the way to escape the apoptosis. The fourth one is limitless replicative potential. It means, in the cancer cell there will be telomerase activation, so the telomere will not be sorted in every cell division, and so the cell will continuously getting divided, which is known by the name limitless replicative potential. There will be sustained angiogenesis. The sixth molecular basis of cancer is invasion and metastasis. And the seventh one is evasion of host immune response. Our immunity will not work to eliminate the cancer cell. So that is the seventh molecular basis. And the final molecular basis, hallmark of the cancer, is growth promoting metabolic alteration, which is known by the name Warburg effect. So that we are going to discuss in our today's lecture in the detail. I have already made the video on self-sufficiency in the growth signal, on the P53, and I have made the video on metastasis as well. You can check the playlist and see that videos. Our today's focus is on the metabolic alteration. So first we will begin with Warburg effect. Now before you understand the Warburg effect, you need to understand this basic cellular metabolism. So every cell in our body needs an energy for doing the work. So they needs the energy in the form of ATP, right? So friends, there could be a two form of cellular metabolism. One is oxygen dependent metabolism and another one is oxygen independent. So whenever the oxygen available in the body, it will adopt a mitochondrial metabolic pathway which is known by the name oxidative phosphorylation. It occurs in the mitochondria, and by oxidative phosphorylation there will be production of 36 ATP, right? And so we can say that it's a most efficient system in our body for the ATP production. So it's the most important pathway that will be adopted by our body whenever oxygen is available for the production of ATP. But suppose oxygen is not available in the body, or if it is in the less amount, then oxygen independent metabolism will occur, which will occur in the cytoplasm of cells. And that metabolism is known by the name glycolysis pathway, in which glucose is used. And by the glycolytic pathway, there will be production of only 2 ATP, and there will be production of metabolic intermediate. So we can say that it is a less efficient system of ATP production. But when oxygen is not available, body has to adopt the glycolytic pathway. So I want to say that whenever there is a less oxygen in the body, there will be shift of oxidative phosphorylation to glycolytic pathway. The body will adopt glycolytic pathway when the oxygen is less. That is known by the name anaerobic glycolysis. When oxygen is not available, the glycolytic pathway will occur, and that is known by the name anaerobic glycolysis. Now our story begins. Now we can understand what is Warburg effect. So friends, interestingly, this particular scientist, this German man, the name is, Otto Warburg. In the 1930, he observed that, despite of available of ample oxygen, despite of availability of the oxygen, cancer cell adopt a glycolytic pathway instead of mitochondrial oxidative phosphorylation. Understand? So I want to say that even in the presence of lots of oxygen, cancer cell doesn't undergo oxidative phosphorylation, and they will adopt a different pathway, that is oxygen independent glycolytic pathway. Even in the presence of oxygen, they will adopt a glycolytic pathway which is characterized by utilization of glucose. There will be utilization of glucose, and there will be increased conversion of glucose to lactose via glycolytic pathway. So, in spite of presence of oxygen, the cancer cell will adopt a glycolytic pathway, and that is known by the name aerobic glycolysis. And that aerobic glycolysis given the name by this scientist Warburg effect. So, Warburg effect means aerobic glycolysis, means glycolysis even though the presence of oxygen in the body. So, that is Warburg effect. And because of glucose utilization, lactate also will be produced. And this increased glucose uptake will occur by increased GLUT1 receptor. This particular receptor will increased on the cell, and they will do the more uptake of glucose. All right. And via glycolytic pathway along with lactose, a lactate is also produced, and this lactate will reduce the pH in our body. All right. Now let's solve one question. Is Warburg effect cancer specific? I mean, it is a cancer specific property. So the answer is no. It's not a cancer specific property. It can be seen in any rapidly growing cell. But what happened in the normal cell? If oxygen is not available and aerobic glycolysis occurs, it will stop immediately once the oxygen available in the body. It does not constantly undergo glycolytic pathway. But in the cancer cell, even though the oxygen is present, the glycolytic pathway will not stop, and there will be constant aerobic glycolysis. Even in the presence of oxygen, the glycolytic pathway will continue, which is known by the name Warburg effect, and which is also known by the name metabolic reprogramming of the cancer cell. So although it's not a specific of cancer, but it constantly seen in the cancer cells. Understand? So I want to say that any rapidly growing cell can show such property, but it's constantly seen in cancer. All right. Now you might have questioned that the oxidative phosphorylation produces a 36 ATP and glycolytic only 2 ATP. Then why cancer cell adopt Warburg effect? Why it is constantly seen in cancer cell? Why they need glycolytic pathway? So here is the answer. Let's understand that cancer cell are very rapidly proliferating cell as compared to the normal cell, right? They are rapidly dividing, so they obviously need a more structural component like that of protein, lipid, DNA, etc. And such structural component cannot be provided by oxidative phosphorylation. For such structural component production, they need a metabolic intermediates which can be available only by glycolytic pathway. That's the one reason why they adopt a glycolytic pathway, because in the glycolytic pathway there will be production of metabolic intermediate which will help in production of structural component of the cancer cell. The second reason is that via glycolytic pathway there will be production of lactate, so the lactate will decrease the pH, and because of reduction in the pH, the tumor can spread easily into our body. Metastasis will be easy. So because of these two reasons, the cancer cell adopt a Warburg effect. So aerobic glycolysis, Warburg effect seen in the cancer cell. That's the answer. All right. Now, what's the clinical utility of understanding the Warburg effect? I mean, what is the clinical implication? So there are two clinical implications. One is diagnostic and second one is therapeutic. First of all, let's see the diagnostic utility of Warburg effect. So we know very well that tumor cells adopt a glycolytic pathway. They will use the more glucose, so they are having the glucose hunger. So that property will be utilized in the PET scanning, positron emission tomography scanning. If the patient will injected a glucose, particularly 8-fluorodeoxyglucose, if injected into body, and if the cancer cells are present into our body, so they will consume a more glucose. So we can say that if tumor is present in the body, more glucose will be utilized and they will be PET scan positive. So the tumor cell and rapidly proliferating cell are PET scan positive. Understand? So PET scan can be used in the cancer diagnosis. If PET scanning is positive in your report, then cancer could be possible into our body. So that is the diagnostic utility of Warburg effect. The therapeutic utility is there. Cancer cell adopt a glycolytic pathway. So you can give the chemotherapeutic drug which inhibit the glycolytic pathway. Alright, so that's the two clinical implication of Warburg effect. Now I'm going to teach you a three important pathway that operate in the Warburg effect in the rapidly dividing cell. This particular diagram is being taken from the Robbins book of pathology. This is a very beautiful diagram that explains the all three pathways that operate in the Warburg effect. So first of all, in the non-cancer cell, in the quiescent cell means they are non-dividing cells. They are not rapidly dividing cells. So in such cell, the lipid and amino acid metabolism occurs via Krebs cycle, and they will produce the ATP by undergoing a Krebs cycle. Understand? And the glucose uptake is not increased. And suppose the cell is not getting enough energy, if there is a starvation, then autophagy will be operated and the cell will eat its own component and they will be reutilized for the production of structural components. So that is the mechanism operate in the quiescent cell. But let's understand how the three important pathway operate in the growing cells. So first of all, let's understand the first pathway, that is a PI3-AKT pathway. So in the rapidly dividing cell, in the cancer cell, this particular pathway is operated and there will be upregulation of the PI3-AKT pathway. And this pathway, will increases the glucose uptake via the glucose transporter. It upregulates the glucose transporter activity, and so more glucose will be taken up by the cancer cell. They will increases the glycolytic enzyme as well, and because of which there will be increased glycolytic intermediates. And these intermediates, responsible for the production of DNA and RNA and various amino acids. It will produces a mitochondrial metabolites as well, which will help in production of lipid, which is known by the name lipogenesis. And this particular pathway, will stimulate a factor that is required for protein synthesis as well. So we can say that, via PI3-AKT pathway, there will be more protein synthesis, more nucleic acid and amino acid synthesis, and there will be more lipid synthesis. So ultimately it will help in cellular growth. So that is the first pathway that is operated in cancer cell. The second pathway that is operated is via, tyrosine kinase receptor. Okay, so via tyrosine kinase, the cancer cell inhibit the enzyme pyruvate kinase, which catalyze the last step of glycolysis. So it inhibit the pyruvate kinase. So because of that, there will be creation and damming effect that leads to buildup of glycolytic intermediates. There will be more and more glycolytic intermediates, and so that it will be utilized, as we can see in this diagram, so that it will be lead to production of more and more DNA, RNA, protein, and the lipid synthesis. So we can say that via tyrosine kinase receptor pathway, also there will be more production of nucleic acid, amino acids, and the protein, which will help in cell growth, which will help in cancer cell growth. The third mechanism is via MYC pathway. This particular pathway is a pro-growth pathway which upregulates the expression of transcription factor, particularly MYC, which is known by the name MYC. And it will lead to, a biosynthesis of many cellular components, and they will lead to a growth. They will lead to, biosynthesis of cellular component by increased glutamine utilization, and that will glutamine will be utilized more by the enzyme. Upregulation of the enzyme name is glutaminase. So in that way, MYC gene also lead to growth of cancer cell. So always remember these three particular pathway operated in the cancer cells: PI3-AKT pathway, pyruvate kinase, tyrosine kinase pathway, and third one is MYC pathway. All these three will lead to cellular growth. Now let's see autophagy and the cancers. All right. Now, what do you mean by autophagy? I have already made the video on autophagy in my cell injury playlist. You can check the playlist and see the video on autophagy. So, autophagy means, self-eating process in which the lysosomal enzyme digests the own cell component in case of an starvation. So, whenever the nutrition is not available to the cell, the starved cell will eat its own component. When the nutrition is not available, they will utilize their own component. They will eat their own component, like that of organelles, protein, and membrane, and they will recycle them for the production of energy. So we can say that in case of an starvation, autophagy mechanism helps in the cell survival. So if it doesn't operate, then cell will die. All right. Now let's see the role in the cancer. So autophagy acts as a double-edged sword in the cancer cell. Let's see how. So as we know, the autophagy is induced by the two important genes. One is bacillin and second one is ATG gene. These two genes are the autophagy inducing gene which act as a tumor suppressor gene, and it prevents the cancer. But in the cancer, if the cancer develop in the body, they corrupt the autophagy process to provide the nutrition for the growth and survival. So by corruption of autophagy pathway, cancer cell will survive and will do the growth. Now let's see exactly opposite. Suppose if the tumor cell have autophagy property, usually the tumor cell doesn't have autophagy property, they corrupt the autophagy process. But if they would have autophagy property, complete autophagy property, then such tumor cell can become dormant, can become non-dividing, and which is a state of metabolic hibernation that allow the cell to survive for the in the hard time for the long period. Why I'm telling that? you know because they can escape the chemotherapeutic effect. How? So friends, understand that chemotherapy will work only if the cells are dividing in the rapidly dividing cell. But if the tumor cell have autophagy property, then they can undergo a metabolic hibernation state, that is a dormant state, non-dividing stage. And in the non-dividing stage of cell, the chemotherapy cannot work. So that could be beneficial for the cancer cell if they would adopt autophagy property. All right. Now the third important metabolic alteration in the cancer, that is the oncometabolism. It's a very new emerging mechanism of the oncogenesis. Here there will be mutation in the enzyme that participate in the Krebs cycle. Particular example is isocyanated dehydrogenase. If it is mutated, if any of the enzyme of Krebs cycle is mutated, then it is known by the name oncometabolism, and it will lead to cancer production. So let's understand the pathogenesis of oncometabolism. So suppose there is a mutation in the isocyanated dehydrogenase, then there will be a mutant isocyanated dehydrogenase will be produced, and such mutant IDH will convert the alpha ketoglutarate into a 2-hydroxyglutarate, which is a oncometabolite, and it will inhibit many epigenome regulatory enzymes. Particularly, it will inhibit a TAT2 family. TT2 TAT2 family will be inhibited, and which is involved in the abnormal pattern of DNA methylation. It will lead to abnormal DNA methylation, and because of abnormal DNA methylation there will be epigenetic change, and which will lead to misexpression of the cancer genes. And because of the cellular transformation is occur, which will proliferate and will lead to cancer production. So that is the mechanism by oncometabolism, right? All right. So this is all about the growth promoting metabolic alteration. So we have seen the three particular or metabolic alteration in the cancer cell. One is Warburg effect, second one is autophagy, and third one is oncometabolism, among which most important one is Warburg effect. Hope it will make your fundamental clears. Now, if you have any question, then you can ask in the comment section. I will answer it in my convenient time. Thank you very much, and I will be right back with a new video. Till then, take care, friends, and bye bye. Thank you very much.